Hello, sir.
Hello?
Hello, sir. Can you hear me?
Yeah, I can hear you.
Yeah. Give me a second, sir.
Can you share your screen?
Yeah, give me a second.
Hmm. Hmm.
Hello. At
Make him co-host. How?
Make him go.
Okay.
This year only.
Hello, Bora sir.
Yes, sir.
Is it can you share the screen now?
No, I cannot share the screen. It's in, it's given permission to you, so you need to allow me to
Permission,
share.
right? Na?
Yeah,
Give the permission. Just 1 second. Be line, yeah.
yeah.
Bora sir. Now you can share now.
Give me a second. Again, sir, saying same thing. Can I rejoin? Maybe now you're given the permission, so I will disconnect and rejoin again.
Yeah, rejoin again.
Yeah. Roy sir, now can you hear me?
Yeah sir. आप try कीजिए.
Yeah, try करा दे.
Now you are the host. Now you try it.
Give me a second.
And now it is visible. Now make it a presentation mode. You can start, I think. You can start, sir.
Now you can see
Oh,
my
wait,
screen.
sir, please, sir. So, good afternoon once again, all the delegates and the participants, whoever joined uh, in our two-day internet symposium. Let us invite. Let us invite our speaker, eminent speaker, as well as our session chairperson. Now I will hand over this session to Dr. C.S. Supriya, madam. She will introduce our today's speaker. Please. Yeah, 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 yeah.
Good afternoon to one and all. It's my pleasure to introduce today's afternoon resource person, Dr. Raghavendra Bohara. And I would like to introduce Dr. Raghavendra Bohara. Dr. Raghavendra Bohara is a biomedical scientist with over a decade of post PhD experience in biomaterials, nanomedicine, and translational medical device research. He currently serves as senior scientist at Cran Medical Limited, Galway, Ireland, and as international adjunct faculty member. His work bridges fundamental nanoscience with the clinical and industrial translation, particularly in drug delivery system, magnetic nanoparticles, glyco-nanomedicine, and biomaterial-based therapeutics. Dr. Bohara has authorized more than 70 peer-reviewed research articles, 13 review articles, 3 edited books, and multiple book chapters with the H index of 37 and over around 6,000 citations. He has been recognized among the top 2% scientists worldwide, and he is the managing editor of Biomaterials indexed in elsewhere and serves on editorial board member of multiple international journals. A receipt of prestigious Irish Research Council postdoctoral fellowship, his research has focused on targeted nanotherapeutics, including selective bone marrow targeting strategies in multiple myeloma and multifunctional nanoparticle system for cancer therapy and the biomedical applications. His work spans protein nanoparticles, liposomes, magnetic nanomaterials, and the translational biomaterial development. Dr. Bohara has extensive teaching and mentorship experience at postgraduate and doctoral level, having supervised around different MSc and the PhD candidates and contributed to the curriculum development in nanobiotechnology and the biomaterials. His translational research integrates laboratory innovation with regulatory scalability and commercialization consideration. And his present, I would like to invite Dr. Raghavendra sir for delivering his session on protein-based nanoparticles and their clinical and translational potential in modern drug delivery. So welcome, sir. S- uh, Sir, I think you have muted your mobile.
Uh, I'm audible now.
Yes, now you are audible, sir.
Yeah. Thank you. Thank you very much, Dr. Supriya, madam. I think it was
Yes, sir.
too long. Uh,
Thank you, sir.
Thank you very much for management allowing me to give a presentation here, especially my friend and uh, the convener of the conference, Dr. Roy sir, and the whole management team from the college. They have given me opportunity to be a part of this wonderful event for 2 days. I was taking the feedback from Roy sir now and then, and he says that there is an enormous response to the conference. So, uh, yeah, I wish to say congratulate him and the whole management team for organizing such a nice event. And they're organizing now in every year. So I wish them to continue this for the overall benefit of the students, community, and the pharmacy domain. So here, first of all, apologize for delayed start because here it's now early morning in Ireland. It's cold day and some server issues, some software issues just glitch, and it took a while to start. So I am quite aware of my timeline. So definitely, I will try to wrap up this all this in 30 minutes so that the further next sessions or next agendas are not disturbed due to my talk. So very quickly, here the whole purpose of this talk is just to give a glimpse of protein-based nanoparticles for drug delivery, and the whole agenda here to present protein-based nanoparticles because I am the man who works or I have worked previously most of my, I can say, a decade on mostly on inorganic nanoparticles, mostly iron oxide, silver, gold, and all combinations, all hybrid structures for drug delivery, hyperthermia therapy for cancer. But when I moved to my when I shifted my career from academic to industry, what I realized that from a regulatory perspective, from a translation perspective, from a approval perspective, the molecules or the carrier system, if they are more oriented to natural uh, design, the regulatory pathways are much more easier. I think I may have a couple of slides there where I can just quickly introduce to the audience. Some of them might know, but definitely that will really help if someone is really trying to drive on with similar research. I think they can really take some important notes from here that selecting the carrier is really essential because what we think as an academician or as a scientist, FDA may not think from that perspective because FDA is thinking from the broader audience or the broader targets, the impacts and all those things. So maybe I can try to introduce. Yeah, moving ahead here now. What is it? Okay, so now I am based in Ireland, and now Ireland is now known to be a global hub for mostly medical devices, and we all know that now it's now in top five countries in world where most of the medical devices, including stents and all other catheters, catheter coatings, are manufactured in Ireland, and there has to be some reasons, and the reasons are in my next slide, where it has shown to show his leadership, global leadership in medtech segment, because uh, Ireland has created a strong industrial base, and out of 25, I think 18 medtech companies are operating in Ireland. And if I want to name few, it's Medtronic. We all know they are the leaders in catheters and stents and mostly interventional procedures. Boston, again, leaders and pioneers in stents. Abbott in medical device diagnostic kits, Stryker in orthopedic implants, including some of the emerging companies, including my company, CrunMed, and some of the more coming in the pipeline, which may you see in coming days. Close to these numbers are need to be updated. Close to 100,000 people are currently working in Ireland, and they are mostly from medtech sector. And why they are coming here? Because we have a really a strong academic industry links, where industry, some of the universities like Trinity College Dublin or there's Athlone University in Galway. They are more industry oriented. The whole projects are industry oriented. The whole PhD work is driven by industry requirements. So there is a whole ecosystem which is developed in the country where the requirements of the PhD, the requirements of industries are considered as a problem for the PhD students. And in this way, the ecosystem is connecting together to make sure that Ireland remains on a top rank in terms of medical device sector. If I put out some numbers, more than 12 billion annual medtech export the country is currently dealing with, and it's one of the Europe largest medical device exporters. And more importantly, the one point which I'm missing in this slide is the whole ecosystem which is funded by Enterprises Ireland to make sure that if someone starts to if someone thinks to start the organization or a company in this on this land, the Enterprises Ireland, which is the governing body, is such a helpful system that just motivates people and provides good amount of seed money to initiate the organization or the initiate the medical device so that it can really turn around. If you have an innovative idea, here the Enterprises Ireland is present to give you such a big amount of money or seed money or some sort of startup initiation so that you can really end up into the large organization, or you can develop your prototype and eventually you merge into the big companies, and that is where the whole business is currently there in Ireland. Okay, now let me come to me. This was the interesting slide. So some of them might know, some of them might not know. I started my career as a medical representative and now I'm working as a senior scientist. This is a career which is all ups and down. So to begin with, I was a diploma. So I started my career with diploma in pharmacy. Immediately I worked as a medical representative for one of the pharma company. I stayed with one company. That's a good part of mine. Worked for 8 long years. Got a couple of promotions there. Work around in Kolhapur and nearby Kolhapur regions. I learned medicine from field doctors and patients. So I can say it was early days where I started to understand what are the problems. May not scientifically that strong to solve, but I was trying to get a flavor of what problems are there in the field. Uh, Further after pharmacy, I decided to work as a in a field, and then after some years, I decided to get back to academics and completed my BSc and MSc in biochemistry from Shivaji University, Kolhapur. Completed my PhD in nanomedicine, which was a funded PhD, and then I started to drive on with postdocs. Got opportunity in couple of different countries. Moved to Ireland. Worked as a senior postdoc in Kuram, which is again a medical device center in University of Galway for 6 long years, and then shifted to Cranmed as a senior scientist. So a journey which is not simple, but it's eventful, it's impactful, it's full of learning. Now, coming back to the topic. So, what I learned and from my previous exercise and what I'm keep trying to learn and trying to understand more, why the drug delivery still fails, because we do have really uh, absolutely magic bullets, but still in cancer or in more chronic elements, chronic disorders, the drug delivery challenges are still pursuing. So what are the main reasons? And if I want to jot down 5 main reasons: this poor delivery, but not poor potency, low bioavailability, rapid systemic clearance, off target toxicity, and poor tumor penetration. So what is the bottleneck here? The bottleneck is delivery, and I think you can see in my image where I have tried to show, may not be clear, that I need to make sure that the images are more clear. Okay, so you can see here with the conventional chemotherapy, uh, the tumor are getting drugs, but the amount of drugs is causing damage to healthy cells and normal and tumor cells is equally impacting to the most of the side effects. And there is no way where tumor where the current chemotherapy is trying to induce the host immune system to kill the cancer cells, and this is the whole purpose why this protein or these nanoparticle based systems are trying to evolve, because apart from killing, they also trying to induce the host immune system cells by just down regulating some of the key receptors, including NKG2D, and ensuring that the immune system gets activated and kill the cancer cells. Nothing new here, we all know. We all know what are the chemotherapy side effects. I won't read all those things, but I will read key bullets: non-specific distribution, high systemic toxicity, dose limiting side effects, and over the period, development of resistance. I think these are the main side effects which we all know and we are learning. Nothing new here as well, because why we are using nanoparticles and types, because being a size from 100 to 1000 nanometer, they can give us opportunity to improve drug solubility, protect drug from degradation. They can give us a, a, a lever where we can use them to control the release and enable to use them for targeted drug delivery. And if I want to categorize them, I can categorize them in polymeric nanoparticle, inorganic, sorry about inorganic nanoparticle, and lipid based nanoparticle. And what I was saying, that my whole research was mostly on inorganic nanoparticles, where I'm trying to slowly move towards protein based or more natural carriers, where the regulatory pathways are much more easier to bring down into market. If so, why I haven't recommend, or why inorganic nanoparticles are staying behind? Because the main reasons for them is not their applicability, but the main reason behind them is they are not easily biodegradable. And when I speak from a regulatory perspective, FDA or any European agency or any regulatory agency, what they want is biodegradability. Even the system is not giving you an benefit, but it should not cause some side effects, or it should not remain in the body and create a toxic effects. So no doubt, if system is not equally effective, or if it's not effective, it's fine, but should not create some toxic effect. And that's the whole purpose of regulatory. And what I feel, that this is the main reason where biodegradability, because being their inorganic nature, there is no way of mechanism where the body can get rid of them, and that's why they are falling off. And some of the really nice inorganic material like gold, silver, iron oxide, where gold can really give us a whole range of stimulation under plasma resonance, and you can create a different size of gold nanoparticles ranging from 2 nanometers to 200 nanometers. That much amount of versatility gold is offering. But the question on degradability is such a big, where FDA haven't approved anything, or may not approve down the line, till this pathway of biodegradability is going to get clear. So why protein based nanoparticles are preferred? The main reason, what I feel, they are biodegradable and safe, and due to which they gives reduced side effect. We can always add couple of more levers to make sure that they can be controlled and provide us a sustained release. And by giving another lever of stilt coating, we can make them prolonged for circulation time, so they can remain available in blood circulation for longer period of time. That's why I think the whole purpose is they are biodegradable and safe. Type of protein nanoparticles for drug delivery. So I think the mostly widely, I have only jotted here few, but you might know there are many there in the literature. But I think the most important is albumin, collagen, silk protein, gelatin, and lipoprotein. And what I feel that, and what I'm going to talk more on this presentation, is something about albumin. I'm going to give a couple of examples from a real world, how albumin and albumin based delivery system is making an impact, And some of my research, which is on silk protein, not on drug delivery, but it is more on wound healing. A couple of slides just to give an audience that we are also trying to move away and trying to work on a more natural carriers. But if I want to list out, these are the 5 to 6 prominent players in the market, and they are now being uh, talk of the town in terms of application like drug delivery, tumor targeting, phototherapy, and imaging. They may not have all the levers to trigger tumor targeting or phototherapy, mostly phototherapy, but conjugating them with certain phototherapy drivers, maybe like adding some photoresponsive dye like indocyanine blue, make them a prominent player to be used as a phototherapy, or conjugating them with again reactive blue, make them a good candidate for imagining, or conjugating them with some of the still coating like PEG or similar coatings, make them a tumor targeting so that they can remain strong, they can remain long for a longer time in a circulation. uh, Gelatin is again something a new, not a new, but again raising the bars because due to again strong mechanical properties and biodegradation properties. So ultimately, the choice depends upon the safety profile, scalability, and regulatory feasibility. So I would drive on. Okay. Just a quick glance for the audience. If some of them they are really new on the protein nanoparticles, there are several methods where I have collected from a review article where you can use them to manufacture, and all these are industry expandable methods: nanospray, electrospray, emulsification, dissolution method, self-assembly, encapsulation. Encapsulation is getting really crazy because it's now industry scale-up method. These are all industry scale-up methods. When you think of these methods, you are trying to scale up this method. So it's again on bench, it's maybe expanded to industry. Uh, from industry's perspective, some of the notes here that dissolution method and encapsulation method are really handy because the amount of QA QC work you need to do on them are really less, and validations are quite easy with this. They offer a controlled particle size, offer both in situ loading and ex situ loading so that you can be more comfortable from the end application perspective. Some of the physicochemical properties you might want from the this system and size, which is around 2200, or maybe you can go up to 200 to 300 nanometers. Morphology again, spherical shape, but the important point I want to say here is that zeta potential, if the zeta potential values are uh, give me a second. Okay, the zeta potential value, if are they are close to -10 to 10 millivolt, so which is very much less as compared to the colloidal system, but that makes sure that they have a less interaction with the cells, so they can remain for longer time. If you have a higher zeta potential value, what will happen? They will interact with the cell more, and uh, they will form uh, a protein particle complex, and your reticoendocrine system will try to engulf and they will be degraded. But if you have this zeta, and that's why even in my current I have seen in industry, we are trying to keep a zeta potential always between -10 to plus or -10. We are not going beyond that just to prevent detected by your host system. Uh, if I want to give some of the major loading strategies, so I think we all might know because hydrophobic interactions, electrostatic, covalent conjugation, and physical entrapment are some of the commonly used loading strategies for most of the protein nanoparticles, because proteins they definitely they are made up of amino acids, so there has to be some amine group and carboxyl group present in their structure. So how we can interact, how we can use those groups or engage those groups to uh, to uh, load our drug. So if you see here, I have a couple of examples in my next slide, but hydrophobic interactions is seems to be more emerging because in most of the protein, the outer structure or the core is available with leucine, isoleucine, and phenylalanine region, and that's amino acids can definitely bind with the hydrophobic drug and can form a strong interaction, or not a strong interaction, but interaction that can carry the payload and deliver at site. Some of the electrostatic interaction includes the model drug like doxorubicin, which is highly positively charged and which binds to the negatively charged amino acids like glutamate, aspartate, and that's why gelatin is now widely used to deliver doxorubicin. I was just going through some FDA trials, and now currently 16 FDA trials are currently ongoing on gelatin and doxorubicin, which are currently in pipeline. And no surprises if those trials come out and you find a product in the market with gelatin as a delivery system. Covalent conjugations is not preferred because we don't want to have a covalent bond, because covalent bonds is remain to be a strong bond, and again you always need, for any covalent interaction you need mostly an enzyme to break down that bond, and so they are not preferred. Physical entrapment, though they are easy, but the leakage is the main concern. So again, but hydrophobic and electrostatic and physical entrapments are some of the most preferred ways from all the perspective, even from industry perspective, to load the drug in this cargoes. I was saying you paclitaxel, and this is your albumin, and this is your paclitaxel. So the phenyl isoleucine, they bind here, and they form a strong hydrophobic interaction, and that carries them, that drives them in the blood. Uh, Roy sir, do I have a time, or I need to just give me a okay, Roy sir will come back. So I was speaking about this clinical example of protein nanoparticle, and which is Abraxane, which is currently available as a commercial albumin-bound paclitaxel
Yes, sir. You
formulation.
can complete within 5-10 minutes, sir.
Yeah, oh, no problem. Yeah. So again, a powerful example of protein nanoparticle is Abraxane, and now which is currently available in market as an albumin-bound paclitaxel formulation. So we all may know that conventional paclitaxel manufacturing requires chemophore reactions, which cause most of the people hypersensitivity. Abraxane is solvent-free manufacturing, which improves safety and tolerability. And now, slowly, FDA is approving Abraxane for breast, lung, and pancreatic cancer, showing the first approval of a protein-based drug delivery system uh, for clinically validated conditions amongst different cancer. So now this is again a first breakthrough in protein nanoparticles in terms of application. So you can see here, this is a NAB paclitaxel complex around 130 nanometer in size. So what is happening here? So your paclitaxel molecules are binding to albumin around 4 to 14 nanometer, and they are forming as a complex which is counting a size of 130 nanometer in size, and that is completely solvent free formulation, and now this is getting approval for multiple applications. So this is how it should go. And again, I mentioned the interaction is hydrophobic interaction, which again relies that the drug is going to be available at some time in tumor. So what advantages? I'm not going to read all those things, but definitely the infusion time, which is most from the clinical perspective, is 30 minutes from 3 hours, and it gives direct, there is no dependent on passive diffusion of drug, but it is directly carried by albumin receptors which are present in the blood and deliver the drug. And definitely from toxicity profile, it has shown improved tolerability and lesser side effects. So that's why more and more indications of cancer are now recommending paclitaxel with albumin conjugates. So this is an ideal example where we can really think of moving around some like gelatin or silk fibroin or some similar proteins where similar interactions can give us some better profile or better safety profile, so that they can be really converted into the product down the line when you try to do think more on similar grounds. That was the whole purpose, to show all this. So coming back to the FDA guidance for nanotechnology, and this was the latest update, US FDA 2022. And so if you see for nano guidance, so for protein nanoparticles, FDA has standard nanoparticles, but if I say, I have another slide which has been more crisp, but I will want to go through with this details. For inorganic nanoparticles, there is no specific guidelines, all case by case. So if there are no guidelines, it's so difficult. What are the parameters? So that's why I think here when you choose for protein nanoparticles, you always are going to make sure that you need to carry, you need to make sure that certain guidelines are from FDA are regulated, and you are through with that. But in kinds of inorganic, there are no guidelines, so you're always in a trouble at some point. Origin is important, mentioned degradability is important, composition is important, and application is important. Where when you see in inorganic table, synthetic, non-degradable, metal, gold, all these are really creating a problem from FDA approvals. So a pathway for protein nanoparticles is guided by FDA nanotech regulations. Origin, biodegradability, and applications. If they are clear, your pathway is much more easier, and your hurdles in terms of case by case review, no specific guidelines, requires extensive toxicity studies. Origin is important, persistence is important, and in application is always important. So much of regulatory pathways here. And I think now I'm very much close to my presentation. So here just I want to share here couple of my example where we are not currently using uh, protein for drug delivery, but definitely we have started to use silk fibroin, which is again a natural protein with some composites like PVAs, which are again FDA approved polymers for dressing material. So one step ahead. So we all know what are the requirements of wound dressing. I won't go in much detail, but definitely for any of the wound dressing, you need a good mechanical strength and blood clotting ability. And I think when you use polymer, you get a better mechanical strength. But with polymer, again the questions of degradation always remain the important driver from FDA perspective. So protein may not give you that strength, but if they have better profile of degradation, they're always preferred. So we have preferred silk fibroin, and this is a very standard method of purification of silk fibrin, where the silk cocoons, which are natural origin, are removed. You get rid of silk sericin, silk fibrin, which protein is prepared. By doing certain chemical procedures, you get a purified silk fibrin, and that silk fibrin is converted into different shapes. Like you can prepare a sponge hydrogels by adding cross linkers. You can use some casting system to prepare scaffolds, and always you can prepare some methods which I shared previously to prepare some of the nanoparticles or films. We have prepared some silk fibrin PVA films, and we have seen some antibacterial activities in different things. We haven't stopped here. We have seen some of the antibacterial activities in rabbit model as well, and definitely as compared to controls which is commercial, we haven't got much results. But if you see in my this first, I don't know if I can, you can see my arrow, but the 0D, this first column, you can see is improving. So the the next step is, now we want to do the whole design validation for this stuff and ensure that we land up in the GMP grade, and trying to mass up the production so that we reach to the end goal. So, take home messages: drug delivery remain a major challenge in modern therapeutics. Protein-based nanoparticles offer biodegradable, biocompatible, and tunable platforms for drug delivery. They improve solubility, stability, targeting, and therapeutic index. Clinical systems such as Abraxane and more in pipeline has shown real-world impact and translation success. Ongoing research is advancing in protein nanocarrier toward precision medicines and next-generation therapeutics. So, I think the future of pharmacy lies not only in discovering new drugs but also designing a smarter system to deliver them safely and effectively. I think that's all from me. Thank you. If anyone has any more questions, definitely they can ask and I can try to answer them. Hello? Hello?
Hello.
Ah,
Hey, Raghavendra, am I audible?
yeah, you're audible. Srini sir, you're audible.
Yeah. So yeah, as I see covalent bonding, I mean, you are mentioning about the covalent bond interaction.
Mhm.
So now people are more interested towards ADCs, antibody drug conjugates, right?
Mhm.
So do you want to throw a light on what is the best delivery system for ADCs, targeted delivery?
So for ADCC, I think albumin is going to be the best delivery system because you don't see if you go for covalent, FDA will never approve because FDA want release profile. And the first FDA says that if you have a covalent bond, how the release profile is going to happen. Then you need to add some external material to degrade. And if you add something external, you need a further approval for that.
Okay, external covalent.
Yeah. So they say that even your loading is very much less, they are least bother. But they say that if something internal is breaking it down and making it available, they are more happy with that. So your loading is less 10-20%, they have no problem. But even if it is not creating further problems, like you bring some external factors to deliver that, then they are concerning.
So again, you have show. I mean, you have a vast experience in delivery systems. So which material do you prefer for nucleic acid delivery systems? You know, I've been using LNPs, but
Mhm.
do you prefer any others? You want? I mean, you would like to suggest any other systems for nucleic acid delivery systems?
Gelatin.
Gelatin. Okay, so
Gelatin
it's a
is now getting really crazy in FDA. There are 15 trials which are available now.
okay. Okay.
Due to tunability, you can easily make it. FDA has clear one pathways. There are a couple of more products are coming now, so you don't need to do all those trials again because you can just show 510K. 510K means previously someone has done, so I am just going to use them as a model and I'm expanding my product.
So gelatin with nucleic acid.
Exactly, it will just really
Okay.
help for you.
Okay. Yeah. Thank you. Thank you, Raghavendra.
I read some message from Roy sir that I should be audible. I don't know if my voice was audible or not. Look, early morning, weather is 2 degree. Voice was not coming out. There was no tea around. I tried and I read that message very late. Sorry about that. If someone has really felt inconvenience with all those things, I really apologize for that.
So thank you, sir. It was really a one great experience sharing your information to all of us. Madam.
Thank you, sir.
Thank you very
Bohara,
much.
sir. So, Sir has given vast information about the benefits of protein-based nanoparticle preparations. And Sir also explained about the novel techniques for the preparation of protein-based nanoparticles, advantages of nanoparticles, protein-based nanoparticles over synthetic one. And Sir also clearly mentioned about the drug loading strategies of nano-based protein molecules. Thank you, sir. You have given in-depth information about the protein-based nanoparticles. Thank you so much for spending valuable time with all of us in this international conference.
Thank you very much again for giving me opportunity. Thank you very much, Roy sir and management team, Supreme Madam as well, and Srini sir, and I think Balaji sir is also there. So those are my good old friends. So I also take opportunity to say thank you for attending and taking interactive part. And yeah, we'll be in touch. Should I leave now?
So, Dr. Bohra sir, thank you so once again, sir.
Yeah.
On behalf of organizer, I will be forwarding the certificate to your mail ID. Thank you,
Thank you
sir.
very much, sir. Yeah, yeah.